Hello everyone. Welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. Okay, without wasting any time, let's get started. So, coming to the definition, bacterial spores are highly resistant, dormant resting structures formed by certain bacteria, for example, Bacillus, Clostridium, inside parent cell under unfavorable conditions, especially due to depletion of exogenous nutrients. What are exogenous nutrients? Basically, the nutrients they obtain from outside which are necessary for their survival. So it is a straightforward definition. And next, coming to the general characteristics, only some bacteria have ability to form spores, not all, and each bacterium forms only one spore. And since spores are formed within parent bacterial cell, they are called as endospores, and the remaining part of bacteria is called as sporangium. It's like if you consider a bacteria, only a part of it will be covering the spore or forming the spore. So that part is known as endospores, whereas the remaining part is known as sporangia. Next, exact stimulus for the sporulation is not clearly known. However, sporulation usually occurs after a period of vegetative growth due to depletion of nutrients. Consider there is a bacteria, it is taking the nutrients and having its vegetative growth, but after a point of time the nutrient supply have been stopped. There is some problem in the nutrient supply, so now it will undergo sporulation, become dormant, and once the nutrient is reobtained, it becomes again a vegetative bacterial cell. Next, coming to the structure, I have given you the diagram which was given in the Apurva Shastri, so just draw it. It is scoring and you have to draw the diagram, and while I am explaining it, just see the diagram and correlate it will help you. Okay, so a bacterial spore consists of multiple concentric layers arranged from inside to outwards. So first, the central core, it is the innermost part of the core, and it contains DNA, ribosomes and enzymes, and it has low water content. Why low water content? Because the low water basically keeps the metabolic activity very low. Okay, so that helps because it want to remain dormant, so metabolic activity shouldn't be higher, so it basically helps, and it is responsible for metabolic inactivity and dormancy. The next part is the inner membrane, which basically surrounds the core and becomes the cytoplasmic membrane of vegetative cell after germination. It's like in spore, it is inner membrane, but as soon as the spore germinates and becomes the vegetative cell, the inner membrane becomes cytoplasmic membrane. Next, germ cell wall, it lies outside the inner membrane and it develops into cell wall of vegetative bacteria. Then comes the cortex, which is a thick layer external to the germ cell wall, composed of modified peptidoglycan, and it maintains the dehydration of core. As we discussed, in the core the water content is very low, so that dehydration is maintained by the cortex, and it is important for heat resistance because there is presence of dipicolinic acid. Next, spore coat, it is tough multi-layered protein coat, and it protects the spores against chemicals and enzymes. Then exosporium, it is present in some spores, not all spores, obviously the outermost covering, and it may show distinct ridges and grooves. Next, sporulation. So sporulation, it is the process by which spores are formed from vegetative bacterial cells. And I have given you a pictorial representation which is basically given in the Anant Narayan. So the vegetative cell is their vegetative cell is present, then a sporogenesis process happens. How sporogenesis process happens we will see next in this video. So after sporogenesis, there is a endospore formation inside the vegetative cell, and then it disintegrates and free spore is obtained, and that free spore remains till how long the unfavorable condition is present, and once the favorable condition comes, the germination happens. Again, germination process is also discussed in this video in upcoming part, and then there is a growth of spore, and then it becomes a vegetative cell. So this is basically a pictorial representation. Draw it, it is good. Next, steps of sporulation. So basically vegetative cell is present. From that, there is something called as spore formation. It is basically appearance of clear area which gradually becomes opaque. So after that, spore wall formation happens. So basically the future cell wall of vegetative bacterium develops, and then cortex and coat formation happens, and finally the mature spore formation happens. Now this mature spore is fully resistant spore, and parent cell disintegrates and free spore is released. Then coming to the germination, it is the conversion of dormant spores into an active vegetative cell when conditions become favorable. What are the steps? So first there is loss of refractility, and then absorption of water and swelling happens. After that, spore wall sheds, and then emergence of germ wall takes place and elongation to form vegetative bacterium. Then shape, position and size of spores. Now shape, it is either oval or spherical, and position is either central, means the center of bacteria, subterminal and terminal. Terminal is the end of the bacteria, central is the middle of the bacteria, whereas subterminal is between both of them, and it is slightly towards the terminal part, so it is known as subterminal. And relation of bacillary body, relation to bacillary body means how it is related to bacillary body. There is two types, non-bulging spores and bulging spores. Now non-bulging spores are those spores whose the diameter of the spore is either less or equal to the width of bacteria, means they will remain inside the bacteria, or they are equal to the size of the bacteria. That's why they are non-bulging, they are not bulging out. Whereas bulging spores, the diameter of the spore is larger than the width of the bacteria, so they basically bulge from the bacteria. Okay, so example for non-bulging spores is Bacillus, whereas example for bulging spores is Clostridium. Next, sporicidal agents. Now spores of medically important bacteria are destroyed by autoclaving at 120 degrees Celsius for 15 minutes. Now again, autoclave and other sterilants which I have mentioned in this photo, we will discuss that in the sterilization video, because they themselves are 5-marker question, they need little time. So for time being, you just note it down, and in the sterilization part that will be clear for you. So spores of medically important, autoclave at 120 degrees Celsius for 15 minutes. Other sterilants are hot air oven, ethylene oxide sterilizer, formaldehyde, glutaraldehyde and plasma sterilization. Next, demonstration of spores. Demonstration of spore means basically we need to see the spores under the microscope, right? So there is a gram staining method, modified Zn staining. Zn staining is nothing but seal nail staining, and there are some special spore stains also. So gram staining, in that they basically appear as unstained refractile bodies, whereas in the Zn staining you see them as weakly acid fast. Now you may have a confusion that what is acid fast, what is Zn staining, what is gram staining, how the process happens. Again, there will be a separate staining method videos, so there we will go into deep in this topic. For time being, note it down. So modified Zn staining, spores are weakly acid fast, appear red when stained using 0.25% to 0.5% sulfuric acid as decolorizer. For now, you can just understand it like you are using something on the spore which makes them visible under the microscope. Now what is the process? We'll see it later. Then there are some special stains like Schaeffler-Fulton stain and Muller stain. Then coming to the application, first thing is sterilization indicator, that is absence of spore growth after sterilization indicates effective sterilization. You basically do sterilization to remove all the microbes present there, right? Now after sterilization, if again the spore growth is happening, that means you have not done the sterilization correct, so you have to do it again. But if the spore growth, when you check for it, it doesn't happen, that means the sterilization part has been done perfectly, so you can move to the next procedure. So that's why we said sterilization indicators. So example is Geobacillus stereothermophilus, which basically seen in autoclave and plasma sterilizer, they check there, and Bacillus atrophius they check in hot air oven and ethylene oxide sterilizer. Now again, these sterilizer or sterilants, we will see it in the sterilization video. And one more is bioterrorism. So spores of Bacillus anthracis used in anthrax bioterrorism attacks. Anthrax is a disease, even it could be go to fatal. So this was used in USA in 2001, I guess. So what basically happened there was they used this bacterial, anthracis and spores in the post mails, and they sent to the obligatories and all, and while opening the mails they got infected, 5 deaths happened. So that is bioterrorism. So there also it was this spores were used. So that's all about the bacterial spores. I hope this video is useful for you, and if you write the answers like this, you will obviously get the full marks. At least you'll get 4.5 out of 5. So do all those diagrams, and that's it. And if you have any doubt, do comment, and thank you very much. Like the video, and keep supporting. Study well, do well. All the best. Thank you.